The dominoes represent the nerve impulse. Once the nerve impulse reaches the synaptic knob, calcium channels open, allowing calcium to flow in. Calcium then will push the synaptic vesicles down towards the synaptic knob. When the synaptic vesicles reach the end of the synaptic knob, the vesicles fuse and release acetylcholine by exocytosis into the synaptic cleft. Acetylcholine then attaches to the acetylcholine receptors on the motor end plate. Acetylcholine then forces open the sodium and potassium channels. Sodium then rushes in while potassium rushes out. This causes a depolarization, allowing an action potential to shoot down the sarcolemma. Acetylcholine esterase then breaks down the acetylcholine. The action potential then goes down the sarcolemma and enters down the T-tubule. Terminal cisternae of the sarcoplasmic reticulum to the sarcoplasm. Before we get to the next step, we need to describe the pieces of the muscle fibers. First, we start with the whole muscle, which is composed of fascicles and surrounded by a sarcolemma. The fascicles are then made up of myofibers. Lastly, myofibers are created by many myofibrils, which the next step will be happening on. The mitochondria makes ATP. Once calcium leaves the sarcoplasmic reticulum to the sarcoplasm, it will soon reach the sarcomere in the myofibril, which is the basic functional unit of muscle made of thick and thin filaments. This is the thin filament. It is made of tropomyosin, the tropamine complex, which is made up of 3 proteins, troponin, TNI, TNC, and TNT. It is also made up of actin for the thin filament. The sarcomere is also composed of thick filaments. The thick filaments are made of myosin, which have myosin heads that are inactive when they are pulled down, but active when they are pulled up. When the calcium reaches the sarcomere, it goes to the thin filaments and attaches to the tropamine, which pulls the tropomyosin off of the actin. This exposes the active sites on the actin. On the thick filament, myosin heads start at a low energy state. ATP attaches to it and breaks down into ADP and phosphate. This will release the energy and put the myosin heads into an active state. Myosin heads then attach to the active sites on the actin. This creates a cross bridge, and the phosphate then breaks free. The myosin head then pivots back to the low energy state. This then releases the ADP, and while this happens, it is still connected to the active sites on the active and pulls the thin filament over the thick filament, which we will see in a second. From a further look, the sarcomere is composed of different regions made by the thick and thin filaments. First, the sarcomere stretches from each Z-band to Z-band. and in the middle is composed of an A band, which is the dark region, which includes thick and thin filaments. Next is the I band, which is the light region, and includes only thin filaments. Next is the H zone, which only includes the thick filaments. And lastly, the Z discs, which anchor the whole sarcomere. From a further view of muscle contraction in the sarcomere, you can see how the myosin head attaches to the thin filaments. When the myosin head get out of the active state, it pulls back the thin filaments over the thick filaments. This is called a power stroke. This is what muscle contraction is. Then new ATP attaches to the myosin heads, releasing the myosin heads from the active site. Therefore, the muscle contraction is done. When the muscle contraction relaxes, the leftover calcium is pumped back into the sarcoplasmic reticulum using ATP. Calsequestrin gathers all of the calcium and stores it for later use.